Hello and welcome. Today, we're diving into the incredible world of gene editing. For years, CRISPR-Cas9 has been the star player, truly revolutionizing how we approach genetic engineering. Its ability to target specific DNA locations with relative ease promised potential cures for genetic diseases and spurred massive biotech innovation. But like any groundbreaking technology, CRISPR isn't perfect. Concerns arose about off-target edits, accidentally changing DNA elsewhere, and its reliance on cutting both strands of the DNA helix. These double-strand breaks trigger the cell's repair systems, which can sometimes introduce errors, potentially disrupting genes. Getting the CRISPR machinery into cells safely and effectively also remains a hurdle for some therapies. These challenges paved the way for even newer, more precise tools. First up is base editing. Think of it like correcting a single typo in a long sentence without rewriting the whole thing. Base editors use modified CRISPR parts, but instead of making a double cut, they chemically convert one DNA letter directly into another. This avoids the risky double-strand break, offering high precision for fixing diseases caused by single-letter mutations, like certain forms of cystic fibrosis or sickle cell anemia. Taking precision even further is prime editing. This acts more like a search and replace function for DNA. It nicks just one DNA strand and uses a special guide molecule, called a PEG RNA, as a template to directly rewrite the genetic sequence at the target location. Prime editing is incredibly versatile, able to make substitutions, insertions, or deletions with remarkable accuracy and minimal double-strand breaks. This could potentially tackle a wider range of genetic disorders, including Tay-Sachs or Huntington's disease. It's worth remembering that before CRISPR, we had tools like zinc finger nucleases, ZFNs, and talons. These are engineered proteins that bind and cut DNA. While more complex to design than CRISPR, they are well established and still used in research and some therapeutic development today. The innovation doesn't stop there. Researchers are exploring ways to edit gene activity without changing the DNA sequence itself. That's epigenome editing. Others are looking at targeting RNA for temporary genetic modifications. And a huge focus is on better delivery systems, finding safer, more efficient ways to get these editing tools into the right cells within the body. Of course, with great power comes great responsibility. Crucial ethical discussions continue around modifying genes that can be passed down to future generations and ensuring fair access to these potentially life-changing, but likely expensive, therapies. So, while CRISPR truly opened the door to the era of genomic editing, the story is rapidly evolving. Base editing, prime editing, and other emerging technologies are pushing for greater precision and safety. They represent the next wave, promising a future where we can tackle genetic diseases more effectively and safely than ever before. Thanks for tuning in.